So let's begin with the first family. Under Myxoviridae, we actually divided into orthomyxovirus and paramyxovirus. So you should know what members come under each. Orthomyxovirus is the usual influenza that you study about, influenza A, B, and C. Whereas paramyxovirus, of course, if ortho guys had influenza, paramyxo will have parainfluenza. So one of the viruses out of these becomes very, very simple. There's another minor one called Nipah virus, which is very famous after 2018. So remember, parainfluenza and Nipah you have learned. Apart from that, you have to learn MMR. What is MMR? It is measles, mumps, but not MMR में R for rubella. No, rubella does not come under this family. It is respiratory syncytial virus that comes under this family. There's a minor change. Respiratory syncytial virus is now known as human respiratory syncytial virus. Not much of a change, but I just wanted to point it out. So the MMR over here, remember, rubella is not R is not rubella. It is respiratory syncytial virus. And on that note, you should be noting that rubella actually is said to be, earlier it was said to be a kind of a toga virus, but now it is said to be a part of something known as Maternaviridae family. That's the update that we have. It's a part of the Maternaviridae family. So repeating, now let's begin all these one by one. Let's start with Orthomyxoviridae family, that is influenza, and this is how the structure looks like. I hope you all remember that all the RNA viruses are unsegmented. However, you remember there was a family called Bira Bora. Influenza had how many segments? It had 8 segments. Can you count? 1, 2, 3, 4, 5, 6, 7, 8. Yes, the RNA has 8 segments. This you studied in your general virology. So 8 segments are in front of you. And if you look at the outer surface, that is the envelope, you will be able to see that there are 2 proteins. One is HA, that is hemagglutinin, which is too much in abundance, so many of them. And then minor ones that you can see are the neuraminidase. So whenever there has to be entry of the virus is mediated by hemagglutinin, whereas whenever the release of the virus has to happen, that is mediated by neuraminidase. Remember, I'm remembering and telling you again, entry ke liye you have hemagglutinin and neuraminidase is going to be from the for the exit. So whenever a virus comes into our cells, maybe our respiratory cells, it will enter via HA, and if it has to come out of the cell, it will come out via Na. Apart from that, can you see 2 more proteins very clearly? There is something known as the M1 protein that you have all over. You can have something called M1 protein, and apart from that, you also have something known as the M2 protein. I want you to focus on this M2 protein. You know why? Because this is the structure that you're seeing in your microbiome lab. Imagine this virus inside your body. Inside your body, do you need all these different things? No, ma'am. Inside the body, the virus has to divide, replicate, make many copies. All that it needs is apna RNA. RNA hoga, virus can make 1 million copies also. So remove all these. As soon as it will come into your body will say remove all these. So who helps in the uncoating of the virus? Who is going to help in the uncoating of the virus when it enters into our body? It is this M2 ion channel. This helps in the uncoating of the virus. So you will say, ma'am, if you give a drug which will not let M2 work, अगर M2 नहीं work करेगा, if this will not work, the virus will not uncoat, and if it will not uncoat, it will not be able to replicate in my body. Exactly, we've got drugs. Amantadine and rimantadine are drugs which are against M2, and that is what you have to know for the treatment of influenza. So repeating once again, if this structure is given in a life cycle, lots of these molecules, yellow color molecules that you have, these are hemagglutinin for entry. These green color molecules are neuraminidase for exit. Then you've got 2 more proteins called M1 as well as M2, and I've already told you the M2 ion channel is the one that is going to help in the uncoating of the virus inside our body. So this is your basic. Now let me show you a twist in the situation. Look at this, guys. What can you see? Yes, ma'am, this is one influenza virus. For example, this is virus number 1, just taking as an example. This is another influenza virus. I will label it as 2. They enter into the same host cell. For example, let's take the example of some animal like pigs. So pigs mein kya hua? Influenza type 1 virus also went into the respiratory cells. Influenza type 2 virus also went into the respiratory cells. Can you see a new virus has only formed? A third type of new virus has formed, which if you look carefully, so it has a little amount of RNA of virus number 1 and it has a little amount of RNA of virus number 2. So you'll say, ma'am, ek naya virus ban gaya. There is a new virus that has has formed, and this is known as antigenic shift. So please note, what is antigenic shift? Where exchange of RNA segments would occur. Exchange of RNA segments would occur, little bit of RNA from one virus, little bit of RNA from the other virus, and a new virus is formed. That is what all of you also do. Little bit of micro you will study from one source, little bit of micro you will study from another source, and then you will come out as a new hybrid variety having all the information. That is known as antigenic shift. And S for S, antigenic shift is said to be a sudden change, and this is associated with influenza A. In fact, all the epidemics and pandemics of influenza that you hear about is because of this. Why are we so scared of influenza? Like you were very scared of COVID, now why are we scared of influenza? Because it can undergo these sudden changes. Mix and match hoga, a new virus will form, it will cause all epidemics and pandemics. Primarily they are caused by influenza A. Versus what is antigenic drift? Antigenic drift is, ma'am, kuch mix and match nahi hai. This is one virus only. Either you can change its hemagglutinin or you can change its neuraminidase. So if you end up changing any one molecule, chota sa change karoge, it will convert into a new virus. That's about it. So that is known as antigenic drift. Antigenic drift is either a mutation in hemagglutinin or neuraminidase. It can be seen in any influenza, but it is not going to cause pandemics and epidemics and all of that. It is not associated with any of those features. An antigenic drift is very, very slow. How I have learnt it is, antigenic shift, S for S, it is very sudden. Whereas antigenic drift is dheere dheere. It is very, very gradual that it's going to be there. These are the two variations that you have to know. This is important in PSM also. Now on that note, just imagine, you want me to mention how would a patient of influenza come? I don't think so. Because we all know fever. We've all had influenza sometime or the other, fever, headache, sore throat, cough, muscle aches and pains. But what is usually absent, what you need to learn in this entire story is that running nose is typically absent. That is important for you to know. Running nose in these patients is typically absent. These are all upper respiratory tract problems. Can it complicate and go into the lower respiratory tract? Yes. As a complication, pneumonia is also known to occur with influenza. How do you do the lab diagnosis? Sample क्या लोगे? Common sense, ma'am. जैसे COVID के लिए you were taking nasopharyngeal swabs. For influenza also you will take a nasopharyngeal swab. And we do the same story. We do antigen detection, antibody detection. We do polymerase chain reaction. That we are doing for every virus. Now, what is so special about this virus? This virus shows a test called as hemagglutination inhibition test. Just learn the name of this because it is very, very important. It might be written as HAI, heme agglutination inhibition test, is going to be positive in a case of influenza virus. That is the influenza ka pathogenesis, lab diagnosis. What about the treatment? I think regarding M2, I have already told you, you can target the M2 ion channel by amantadine and rimantadine. But what can you usually use? Do we use Tamiflu when you remember that swine flu outbreak and so many influenza outbreaks? So we use something called Tamiflu, which is oseltamivir. O for oseltamivir, O for oral route. This is a drug which is given orally. Apart from that, there is a drug called zanamivir and Relenza. दोनों में क्या आ रहा है? Neza वाला sound है. So this is going to go via inhalation or the nasal route. Easy to learn हो गया. Zana sounds like Neza. Relenza sounds like Neza. So this is going to go via nasal route. Whereas oseltamivir, which is Tamiflu, oseltamivir is going to go via the oral route. And that wraps up your Orthomyxoviridae family. Now I'm going to come into Paramyxoviridae family. Number one, we had something very similar called parainfluenza and Nipah viruses. These were the chota wala viruses that you had. Then you have measles, mumps, and not R for rubella, respiratory syncytial virus. So let's start. Let me start with the major ones. Measles and mumps main wale hai na. Let us finish off those. Measles was a recent FMG and a NEET PG question also, so you have to know measles. Measles is something known as rubeola. Ab na there is all-time confusion. What is rubeola? What is rubella? What is German measles? Let me figure it out once and for all. There is something known as measles. and there is something known as rubella. ये तो पता है, measles and rubella. So अब rubella is not, you can't put another rubella into it. So this is what is called as German measles. This is referred to as German measles. And measles is referred to as rubeola. So cross connection हो रहा है, ठीक है? तो measles is referred to as, तो cross connection type याद रखना. एक बार एक ही measles आएगा, एक बार एक ही rubella आएगा. So if measles is written, don't write German measles, then write rubeola. If rubella is written, then write German measles. One thing to note. Other than that, please note that their incubation periods, I am telling you measles, mumps and rubella ka incubation periods ek saath. So please note that when you are talking about, or better than that, what I can say is I'll give you a table. Why only incubation period? Let me just finish off all of the vaccines also, because then pediatrics is done. Let's talk about measles, mumps and rubella. So for measles, the incubation period is going to be 10 days, right? 10 days as soon as you get a question. Now from the same 10, I want all of you to make a line down, and how many days does it become? For mumps, it's going to become 19 days. Now once you have drawn the 19, from that, please take another danda forward, take another line forward, and for rubella, it becomes 14 days. So repeating once again, this is how you're going to write it in the exam. For MMR, you will write 10 days, then you will convert it into 9, so 19 days, then you will convert it into 4, so 14 days. 10, 19, 14. What about the strains? Remember, for measles, I always, now from today onwards, I will like to call it measles. Okay, we'll write it as Z bana dete. Let's make it measles. Why? Because the strain that we have is going to be the Edmonston-Zagreb strain. So it's the Edmonston-Zagreb strain that we are going to use. So measles mein Zagreb. What about rubella? This is very simple. Ra27/3. So RR rubella, Ra27/3. Mumps. Mumps mein I will just teach you, there's always a chota sa cute sa bacha, that a photo of a chota chubby cheek bacha is there. So that bacha we have named as Jerry. So the name of the vaccine strain is going to be the Geril-Lin strain. So these are the strains that you have for MMR. So remember, for measles, you have the Z, that is Zagreb strain. For mumps, you have the cute Jerry, that is Geril-Lin, and R ke liye Ra27/3. What are the incubation periods? 10, 19 and 14. So this is what we've done. Measles is known as rubeola. Incubation period is 10 days. Now what is the question that came in the paper? This is a recent exam question of 2023 for FMG students. Measles is gonna be the 3 C's. तो क्या क्या होगा देखो मैम, वायरस है, फीवर तो होगा, understood. Other than that, there is cough, conjunctivitis and coryza. Remember, cough, coryza, conjunctivitis. If these 3 C's are written, it is very, very important that you think of measles in the exam. Cough, coryza, conjunctivitis. But what after that? देखो, incubation period क्या था मैम? 10 days था. तो 10 days होते ही, what is the first thing that the patient is going to have? Patient will have fever. Immediately 2 days later, the patient has these spots on the buccal mucosa near the molars. Near the molars, along the buccal mucosa, there is spots which are known as Koplik spots, right? And another 2 days later, we start seeing a rash in these patients. And this rash is very characteristic, because when rash will come, everything else is gonna go away. Jab rash hoga story mein, to fever is gone, Koplik spots are gone. So remember, 10th day, that is when the incubation period is completed, fever happens. 2 days later, Koplik spots happen. Koplik spots are seen on the buccal mucosa near the molars. And then we have 14th day, that is rash. And can you see how the rash is happening? As if someone has thrown water on you. Someone throws a bucket of water on your head. First, the water will go behind your ears, and then gradually it will come onto your neck, and then it will go to the body. So first it has gone on the face and behind the ears. It starts behind the ears. Then it's going to go on the entire shoulders, the neck area, and then it's going to go on the trunk. That is how, as if water is falling down. That is the classical rash characteristic. So coming all the way back, when we are talking about measles, kaun sa wala spots likha? We have written Koplik spots, because when you go to pediatrics and when you study rubella, rubella also has a kind of a spots, and those are known as the Forsheimer spots. They are present on the palate. Forsheimer spots are present on the palate, and we will be wanting to know these differences in the exam a lot, because dono mein fever hoga, right? Measles ke के साथ कॉप्लिक स्पॉट्स लिखा होगा, रुबेला के साथ फॉर्शाइमर स्पॉट्स लिखा होगा. ओके, कमिंग बैक टू मीजल्स. तो मीजल्स का क्या हिस्ट्री होगा? देयर विल बी अ कफ, कोराइजा, कंजंक्टिविटिस, 3C हिस्ट्री, then fever, Koplik spots and rash. But what is a very important complication that can occur? There is a CNS complication known as SSPE. So what is SSPE? Subacute sclerosing panencephalitis, and this actually is said to be a late complication. When I say it's a late complication, it's going to occur many years after measles. So for example, a patient had measles right now. How many years later will SSPE occur? Approximately 7 to 10 years later. But the problem is that once SSPE occurs, within 2 to 3 years, it's going to be very fatal. It's going to result in death. So please remember, late complication hai, but once it happens, it's quite fatal. Now, one thing I want to ask you from pathology again, integrating it, was there any cell in pathology which reminds you of measles? Was measles associated with any kind of giant cell? M ka ulta W, yes, M opposite inverted W, Warthin-Fink-Elde giant cell. What is Warthin-Fink-Elde giant cell? Something that had intranuclear and intracytoplasmic inclusions. If you remember this, it had intranuclear. Can you see dots inside the nucleus and also inside the cytoplasm? So you had intranuclear and intracytoplasmic inclusions. Any other virus that shows you intranuclear and intracytoplasmic inclusions other than measles, we have studied just half an hour, 40 minutes ago in this lecture series, and that is cytomegalovirus. Measles and CMV show both IN and IC inclusions. Okay, so measles is done. I think we can go on to the next one, the cute little kid. I told you, I will show you a very cute little kid and I call him Jerry, and that is known as mumps. Mumps is what? So obviously, if I ask you what is the most common feature of mumps virus, you will say, ma'am, nothing. The most common feature is inapparent. Most of the patients actually don't have anything. But when they have a manifestation, I think you can see why is this kid so cute, chubby, because he has bilateral parotitis. Bilateral parotid gland involvement will be noted. What is the second most common manifestation? That is going to be unilateral orchitis. So it means there is involvement of testis, unilateral orchitis, and involvement of bilateral parotid. Imagine just if you say, if there's a bilateral parotid gland involvement that is given in the exam and you are given a child, what is the infection that you will think of? You will always think of mumps. But if you've been given an adult and that adult is a smoker, then what is the infection that you are going to think of? Maybe not an infection, but a tumor, and that tumor in surgery is known as the Warthin's tumor. Remember this dictum forever, it will help you a lot. Bilateral parotid gland involvement hai. Children mein I will think of mumps, whereas in adult smokers I will think of Warthin's tumor. That is important. That is what you need to know. Coming back, mumps ke liye, I'm not going to tell you the strain. Why did I call him Jerry? Because the vaccine is going to use the Gerryl Lynn strain. We have already done that. We've also done the incubation period, and that was 10 for measles and uska 19 banana tha. So it was 19 day for mumps. Actually, I always study measles, mumps, rubella, but does rubella come under this family? No, rubella does not come under this family. Still, I am teaching you rubella over here because pura vaccine ho jata hai na. So let me teach you rubella, and I think I have already told you rubella is also now, earlier it was on the Togaviridae family, but now it is said to belong to the Metonaviridae family. I think this information also I have given you, that measles is known as rubeola, rubella is known as German measles. I think I have also told you the spots. So if we study rubella, firstly, what is the incubation period? Again, let me ask you your mnemonic: measles, mumps, rubella. Pehle 10 hoga, then 10 ka 19 hoga, and then 19 ka 14. So what is the incubation period for rubella that we have? We have 14 days. What all does these patients have? First and foremost, it's an upper respiratory tract infection. There is a post-auricular lymphadenopathy. There is a rash that you can see over here in these patients, and you can see some red color, pinkish red color spots on the palate. What are those spots? Fordsheimer spots, and that is what I was telling you. In measles, what were the pathognomic spots? Measles mein most important are Koplik spots, whereas in rubella, what is important? It is the Fordsheimer spots, which are non-specific but are quite important for your MCQ. So that is how you remember measles versus rubella. But If you talk about pediatrics, all this information will just go through wala information. What is important from pediatrics is the rash. This rash tends to last for 3 days. This rash classically lasts for 3 days, and that is why we also call it a 3-day measles. Did you notice in the previous slide along with German measles, I had written 3-day measles because the rash lasts for 3 days. That is a pediatrics question. Another pediatrics question is, can this go from mother to child? Yes, mother to child. Congenital rubella syndrome, CRS, is very, very important. What is congenital rubella syndrome? So rubella sounds like Rona. Number one, Rona. Who is gonna cry? Someone who is blind in love. So remember, प्यार में अंधा बहरा. That person is going to cry. Never go blind in love. ठीक है? तो प्यार में अंधा बहरा means what? प्यार में means that this person is going to have a heart problem, patent ductus arteriosus. अंधा बहरा means that there is a cataract, and बहरा basically means there is sensory neural hearing loss. So remember, प्यार में अंधा बहरा is going to cry, that is rubella. But also remember, Bella will remind you of blueberry muffin baby. So can you see these spots that are present which are giving it a characteristic blueberry muffin appearance? So remember this as well. I'll repeat the mnemonic for you so that you don't get confused. Rubella means Rona. Pyar mein andha behra is gonna cry. So pyar mein patent ductus arteriosus, andha for cataract, and behra is going to be for, we have the sensory neural hearing loss. Next, Bella will remind me of blueberry muffin baby that is formed over here. But one question which I want the INI-CET students to note, in congenital rubella syndrome, do you know the virus? The virus is shed in the saliva and the urine of the baby. So whenever there is a congenital rubella syndrome baby, look out for the virus in the saliva and the urine. That is what is important from INI-CET point of view. So now rubella was not a member of this family, but humne kar liya. But what is actually a member of this family with R, what you actually had to know was respiratory syncytial virus. So break it na. Respiratory syncytial virus, first and foremost, ma'am, it is quite common in R for R, that is the rainy season. Next, it is going to cause respiratory problems. So in children, it is going to cause lower respiratory tract infection. In adults, it causes upper respiratory tract infection. Either ways, it is going to cause respiratory tract infections. That is common sense. Now, what I need to know is what is the meaning of the word syncytium, because please remember, microscopic examination shows you a syncytial appearance. Syncytium appearance means syncytium, like 10 people are studying together, they have made a syncytium. Group banana. Group banana means multinucleation. That is what you notice in respiratory syncytial virus. So the name is enough na. Respiratory, then we have rainy season. Respiratory causes respiratory tract infection. Syncytium matlab it's going to make a syncytium, and that is RSV for you. What is the last member of this family that we are left with? We are left with Nipah virus. I had told you a while ago that, after the 2018 outbreak of Kerala, this Nipah virus became very, very famous. Why? Because this had a mortality of almost 60 to 70%. Why were so many people dying? Because what part of the body does it affect? It affects the brain. It is causing encephalitis. So please remember, when we are talking about Nipah virus is very, very dangerous, or having a very high mortality. It is going to cause encephalitis as well as fever. And how will you identify the life cycle? It is going to be associated with fruit bats. So remember, the bat urine, that is the vector actually, right? The fruit bat is the vector, and the bat urine contaminating our food and water can get the infection to us, number one. So it can come via our food and water. Secondly, we also have pigs that can be involved in the life cycle. So please note, whenever you're talking about Nipah virus, first and foremost, you have to think of pigs. You have to think of pigs, which has to be there in the life cycle. Second, this can also be associated with patient secretions. These can also be associated with patient secretions. And apart from that, you have to learn the fruit bat, which is, so you see that fruit bat wala ek life cycle, you know that, okay, this is associated with Nipah virus. And if you read it opposite, in will remind you of what the patients have. Patients have encephalitis, which is very, very important. Very, very high mortality. There is a high mortality. That is why I had written Nipah separately. Everyone repeat with me. What will be there in the life cycle? Pigs will be there in the life cycle. Can it spread from patient secretions? Yes, it can. If I start reading it opposite, in pada, it is going to be known as encephalitis hota hai. What is the mortality? It has a very, very high mortality, 60 to 70%. And the only extra thing that you have to learn is going to be the fruit bat that is there in the life cycle. That is about Nipah virus, and that finishes off all of your Paramyxoviridae family. What do we come down to next? Next, we take up the Picornaviridae family. If I tell you the classification of Picornaviridae family, honestly, there are two groups, but you don't have to study all. So let us focus on the ones that you have to study under Picornaviridae. Number one, I can see there's a family of, see, देखो कैसे divide करा. Picornaviridae means either it is going to cause some intestinal fecal-oral wala, entero wala infection, or it is going to cause some inhalation wala infection. So inhalation means nose. Rhinovirus, common sense hai. This means rhinovirus is going to cause common cold. Very, very obvious. The only question that I expect on rhinovirus is what is the attachment receptor for rhinovirus, and the attachment is going to be with ICAM1. This is the only question for rhinovirus that we expect. It is going to come and attach to ICAM1. Rhinovirus ho gaya, khatam. Do you remember any parasite that attaches to ICAM1? So ICAM1, if a virus is asked, then which virus is going to attach to it? Rhinovirus will attach to it. If a parasite is asked, Plasmodium falciparum attaches to it. So rhinovirus story ho gaya. Enterovirus. So this will include polio, Coxsackie, of course, enteroviruses and ecoviruses. Out of this, you get a question on polio and Coxsackie, which everyone knows. Polio is something that you study a lot in medicine and in pediatrics, but I'll still give you a quick overview of what all is happening in polio over here. If I ask you what is the basic, matlab polio, jab final polio, you say a patient has polio, before I get into this entire life cycle, I think everyone knows that this is famous for having descending flaccid paralysis. That is the primary thing that polio causes, descending flaccid paralysis. But how many phases does polio go through? So whenever the polio virus is going to enter, so for example, there is some feco-oral route by which the polio virus is going to enter, and if it has gone, matlab it's coming through the oropharynx, and after oropharynx, matlab it has to go to the GIT. If it has to go to the GIT, I will say this is the alimentary phase. So it's gonna go to the GIT. I can see it has gone into the intestine, and it can come out in the feces also. So that is the alimentary phase. But do you see that apart from this, a part of it has also gotten lodged somewhere near the tonsils. मतलब थोड़ा सा अगर oropharynx में है, थोड़ा सा intestine में है. So from there it will go on to the respective lymphatics. So from the oropharyngeal region, which lymphatics do you think it will go to? Of course, it will go to cervical lymph node. From intestine, which lymphatics will it go to? Obviously, it will go to mesenteric lymph nodes. So ma'am, when it has gone into these lymph nodes, call it the lymphatic phase. Now, the next phase into which it enters is the lymphatic phase. From the lymphatics, now it's going to go in, every time lymphatics drain into the blood. So from the lymphatics, it's going to drain into the blood. Something, virus going into blood means now I call it viremic stage. Once it goes into the blood, I'm going to call it viremic stage. And from there, obviously, it is going to go to the different parts of the spinal cord, that is the neural phase. And that is when the patient develops descending flaccid paralysis. Repeating, first it goes into the gut, so alimentary phase. From there it goes into lymph nodes, so lymphatic phase. Lymphatics always go to blood, so viremic stage. And from viremia, it goes into the different parts of the spinal cord and the motor neurons, so that results in paralysis. What sample do you think you're going to take? Ma'am, ek to na, you will say, ma'am, ek to I want to find out because polio has been eradicated and all these things that are being said. So in the community, first I want to find out that is there a risk still present? So I think what you should be doing for that is take a sewage sample. Why do I take a sewage sample? Because I just realized that polio is going to get excreted in the feces also. So sewage sample to lena padega. That is one thing. But if I have a clinical patient of polio, so sewage sample you're taking just to know the burden in the society, right? But if I have a patient of polio, the sample that can be taken again, ma'am, from the patient also. Right now you are saying that it is going to come out into the feces, so take a feces sample. Ma'am, you also said that it's going to go somewhere in the oropharynx, so you can also take a throat swab. These are the kind of swab, either a throat swab or a rectal swab can be taken, but the isolation is very, very rare, as they say, right? So this is what you do. For knowing the epidemiological burden, you take sewage samples. For taking patients, you take throat or rectal swabs. Now, this was about polio. Do you remember, other than polio, what is the other one that I told you we'll be studying? We'll be studying Coxsackie. How many types of Coxsackie do you know of? We know of Coxsackie A as well as Coxsackie B. And I can see Coxsackie has a lot of serotypes. Coxsackie A has a lot of serotypes like 1 to 24. Coxsackie B also has 1 to 6. So, kaise yaad kare? What is common between them? Both of them cause hand, foot, mouth disease. Very simple. There will be lesions on the hand and the feet and the mouth. Hand, foot, mouth disease is caused by both. So Coxsackie A is going to cause H, H and H. What is H, H and H? Number 1, it causes herpangina. Number 2, it causes hand, foot, mouth disease. And number 3, it causes acute hemorrhagic conjunctivitis. Acute hemorrhagic conjunctivitis. They ask you this question. Don't take this question lightly. Acute hemorrhagic conjunctivitis is caused by Coxsackie A, okay, but they ask you which Coxsackie A. तो जो last वाला है, that is Coxsackie A24, which is also known as enterovirus 70, because do you remember, right now I told you Coxsackie is actually an enterovirus. So which enterovirus can it be? Enterovirus 70 is very, very important. That is what is Coxsackie A24, and this causes acute hemorrhagic conjunctivitis. Repeating, Coxsackie A causes herpangina, causes hand-foot-mouth disease, causes acute hemorrhagic conjunctivitis. Whereas Coxsackie B, hand-foot-mouth disease to hai hi, but with B, you will start making a list. And it causes something called as the Bornholm disease. Bornholm disease, एक C छोटा सा लिखो, that I'll come to later. Bornholm disease is also known as devil's grip. It is also known as dynia, pleurodynia, and it is also known as epidemic myalgia. Only important for INI-CET students. Repeating, Coxsackie B causes Bornholm's disease, known as devil's grip, known as pleurodynia, known as epidemic myalgia. That is what Coxsackie B causes. And what is this tiny little C over here? It can also cause cardiac involvement, that is carditis. So please note, these are the differences. But if you say, ma'am, last day, and for FMG students especially, if you ask me, ma'am, what is that one thing in this table that I have to learn? I will say, learn acute hemorrhagic conjunctivitis is caused by Coxsackie A24. That is what you learn. Okay, done with this. We are done with the entire Picornaviridae family, right? Picornaviridae is done.